Hello everyone. This is Dr. Meera Srivastava from Uniform Services University of Health Sciences from Bethesda, Maryland. First, I would like to thank all the organizers for inviting me to present my study on validation of race and recurrence specific biomarkers. Briefly, over the years, we have identified several race and recurrence specific serum candidate biomarkers using protein array platforms with the support of Martha Cancer Center which is situated in Walter Reed Army Hospital in Bethesda. As a brief introduction, racial disparities in breast cancer incidence and mortality have been very well documented in the past, particularly among African American women who have been found to have a lower incidence of breast cancer compared to white women, but 40% higher mortality post-diagnosis compared to white women, a disparity that emerged in the 1980s. This could be due to several possible causes, and that may include differences in disease characteristics at presentation, treatment or delays in treatment, socioeconomic status, presence of comorbidities, or biological factors. The observed differences in the incidence and disease aggressiveness support a potential biological role for different biological factors and pathways of breast carcinogenesis between African Americans and Caucasian women. From the systems biology perspective, the DNA tells us what could happen in terms of genome, and when it gets transcribed and the RNA could tell us what might happen when we study the transcriptome. And when RNA gets translated into proteins, that will tell us what's happening when we study the proteome. And metabolites, which are the end products of all this, what has happened. And when we study the metabolome, we will come to know what has happened. So we attempted to study the proteomics. So proteomics is like one gene. It can give rise to many proteins when it gets transcribed through alternative splicing, which can give rise to protein A, B, C, and then through post-translational modifications, one gene can give rise to many proteins. So we have 23,000 genes, so there is a possibility for 500,000 proteins to come. So the proteomics problem is, first there are 24,000 protein coding genes in the genome, so we know who they are. But with the best mass spectrometry techniques, we can identify 2,000 high abundant proteins, which means you identify 5 to 6 peptides per protein, greater than 10% coverage. So you have to be, of course, beware of one-hit wonders. So mass spectrometry is stochastic. But to see low abundant proteins, we need antibodies. There are more than 10,000 unique antibodies commercially available. So we need antibody and protein microarrays to detect these low abundant proteins. So our proteomic strategy has been to discover the candidate protein biomarkers in patient sera using high throughput antibody microarray platform, and then to validate them using a reverse capture protein microarray platform. So in this antibody microarray platform, you print the antibodies on the slide and then you incubate with serum where the antigens are present, so they will bind to the antibody which we know already what they are. And the reverse capture is just the reverse. You print the antigens in the slide and you go with the antibody of interest, for instance, a candidate protein biomarker. So we used this to first discover and then we validate. So in breast cancer, there is several markers that are identified which have limited impact on bedside decisions such as prognosis, surveillance, and monitoring of disease. Markers for predicting recurrence and metastasis are they are very limited in number and they are low abundant and quite non-specific. So we are faced with this challenge. So the rationale for the selection of biofluids is that uh, we used serum as our sample resource for identifying biomarkers because biomarkers usually they can be directly identified in the tissue at the site of expression. However, harvesting tissue is considered more invasive and they are less well tolerated longitudinally. Since laboratory tests used in the clinical setting are targeted at expressed proteins, biofluid proteomics has become popular in identifying biomarkers, and optimal drug targets as a non-invasive tool for monitoring aggressive breast cancer in a cost-effective fashion. So for complex disorders that affect multiple organs, there is a lot of interest in developing quantitative serum proteomic signatures since blood draws are common in clinical practice. The problem is that serum biomarker, due to the complex biomarker, currently suffers from serious constriction due to the complexity of the serum sample and the technology. Additionally, disease biomarkers are typically low in abundance and statistically significant proteins are still bottlenecks for proteomic analysis. So we hypothesize that proteins that are released into the serum are from the diseased patient's cells and can be detected non-invasively. We can detect the race-specific biomarkers and subtype-specific biomarkers, recurrence and metastasis-specific biomarkers. In addition, response to therapeutic interventions and drug resistance can also be followed by frequent measurement of such biomarkers. So our strategy has been to use proteomic strategy and proteogenomic strategy. So in the first case, proteomic strategy, to discover the candidate protein biomarkers using high throughput antibody microarray platform and validate with reverse phase protein microarray platform. And in the second proteogenomic strategy, we first analyze the data sets from CCLE which is a cancer cell line encyclopedia, and TCGA for breast cancer specific genomic biomarkers, and then validate the top candidates using a reverse phase microarray platform on breast cancer patient and control serum. So we have, this is the table which shows the sample selection for proteomics. We obtained the serum from the Martha Cancer Center. These are 240 patient cases were selected, composed of controls and all immunochemistry based subtypes, subdivided by pre and post-menopausal status and by race. We attempted to obtain 10 samples for each group So the immunohistochemistry subtypes are triple negative patients, HER2 positive and negative patients, and luminal A and luminal B. So we take the serum from all these subtypes and label the disease serum with the Cy3, which is a fluorescent dye, and incubate with the normal serum labeled with Cy5. This will serve as the internal standard. So we mix them both, and we apply that to the antibody microarray. Here you see the sample and the internal standard binding to the same spot on the array, and the unbound antigens are washed away. So then we measure the bound antigen through scanner, and then we did the and the typical antibody microarray, which has 1024 features, is represented here, and the proteins that are elevated in one source compared to other is either fluorescent green, which is downregulated, red, which is upregulated, is shown here, and when they have similar levels, they appear yellow. And we did bioinformatic analysis, and that identified several proteins which are race specific, which are interestingly VEGFR2, a protein linked to breast cancer metastasis and poor prognosis, was significantly overexpressed in AA cancer serum compared to Caucasian, suggesting a possible explanation for the higher incidence of aggressive breast cancer in African Americans versus Caucasian race. Here is another protein, which is, this slide shows the upregulation of CTBP1, which is C-terminal binding protein 1, a transcriptional co-repressor which has been found to promote breast cancer cell proliferation and migration, as well as resistance to chemotherapy. As you can see that the African Americans have higher levels of CTBP1 compared to the Caucasian race. This slide, then we did a phosphospecific antibody array, which was specific for NF-kappa B signaling pathway, and we found that we identified top proteins in Caucasian patients which were upregulated. For example, here is ubiquitin. The green is the Caucasian race and the blue is the African Americans. As you can see that ubiquitin, NF-kappa B, P105, and the serine 927 represents, that is the site where it is phosphorylated. And you can see upregulation of I-kappa B alpha, NF-kappa B P65 at threonine 254 site, and NF-kappa B P65 at phosphorylated at serine residue 536. So these were differentially regulated, these biomarkers, between African Americans and Caucasians. Then we validated using the reverse capture lysate microarray. So what we do is we print serial dilutions of serum samples from many patients are prepared using a robotic diluter in 385 well plates, and then they are arrayed on multiple slides by a microarray printer. And the slide is consists of nitrocellulose baked glass slides, and then they are probed with different antibodies. Here you can see the dilution series going down. This RPPA, we call it reverse phase protein microarray, provides an efficient platform for large-scale quantification and validation of proteomic differences and biomarkers. We can print 1,000 patient serums on one slide. And then we obtained recurrence specific samples out of the 240 samples after 3 to 4 years. Some of the patients recurred. We had 21 samples, and most of them turned out to be triple negative here. This is African Americans, this is Caucasian race, and HER2 positive. Then we did the antibody microarray and reverse phase to confirm. We see that we identified several epigenetic specific biomarkers such as HDAC9, C5, and C7, and also we identified and you can see the AUC value for C9 is 0.864 and C5 is 0.77. And we also identified using mesoscale discovery platform. There are inflammation-related proteins such as serum amyloid protein A and IL-18. They were upregulated in recurrent patients compared to non-recurrent patients with p-value of 0.003 and AUC of 0.79. So our second strategy was to do the proteogenomic strategy, and we analyzed data sets from CCLE and TCGA for breast cancer specific genomic biomarkers, and we validated using the reverse phase microarray. And we identified vitronectin, XOSC4, TGF-beta receptor 2, and IL-8 as race and recurrent specific biomarkers. And we did some cell line experiments with the breast cancer cell lines. We came to know that they activate the PI3 kinase AKT pathway. This is one example of how we analyzed the vitronectin. Here we had sample data for survival and copy number alteration for 2,173 breast cancer patients. And as you can see that the high copy number variation of vitronectin has bad prognosis, means less survival compared to the blue. So we analyzed other candidate biomarkers which were having differences in the survival outcome. And then we take this and did the reverse phase microarray using our sample set, and we got the same results of recurrent specific, race specific recurrent biomarker was indicated by the vitronectin levels, higher levels. And this one is XOSC4. You can see that the higher level is present in the recurrent patients in comparison to non-recurrent, with AOC value of 0.68. In summary, VEGFR2, a protein linked to breast cancer metastasis and poor prognosis, was significantly overexpressed in African American cancer serum compared to Caucasian patients. We have identified several key epigenetic biomarkers such as HDAC5, C7, and C9, and inflammatory biomarkers like serum amyloid A and IL-18 and NF-kappa B as recurrent specific biomarkers. These top two were obtained using the antibody microarray platform and confirmed by reverse phase microarray. However, the proteogenomic data identified betronectin, XOSC4, TGF-beta receptor 2, and IL-8 as race and recurrence specific biomarkers activating the PA3 AKT pathway. And these race-specific biomarkers that discriminate between recurrent and non-recurrent patients have a ROC value between 0.7 and 0.86. So the significance is our study yielded race-specific breast cancer recurrence biomarkers, which can be used as a cost-effective quantitative tool for multiplexed detection of disease progression, leading to a rapid translation of the findings immediately to clinical use. Additionally, it will also provide enhanced capabilities for risk stratification and monitoring of recurrence, opening the door for personalized Medicare. Thank you very much, and I am open to questions.